I'd like to welcome everyone back to the first annual Unither conference on nanomedical and telemedical technology. And we're now picking up with the keynote speaker for our first day, Dr. Baruch Blumberg, who very kindly agreed to substitute for Ray Kurzweil, who was unable to be here at short notice. But fortunately for us, as indicated in my introduction slide, which was actually done before I even was aware of the substitution, Barry has been operating at the nanoscale for many years for great human benefit and fantastic international renown. Dr. Blumberg is currently a distinguished scientist at the Fox Chase Cancer Center, and he's also the chairman of the scientific advisory board of our hosting company, United Therapeutics. He has served as director of the NASA Astrobiology Institute and as principal scientist at the NASA Division of Fundamental Space Biology. Dr. Blumberg has also for many years been a senior and valued member of the National Institute of Health Staff. Barry was awarded the Nobel Prize in 1976 for discoveries concerning new mechanisms for the origin and dissemination of infectious diseases, specifically the discovery of the Hepatitis B virus. Dr. Blumberg is also a real pioneer in the field of field biology and has traversed some of the most interesting places on the planet Earth. Dr. Blumberg has two lectures that are available to share with us today. We are only guaranteed to get one, which is the lecture concerning the discovery and the sequelae therefrom, the fascinating sequelae therefrom of the hepatitis B virus. But he also has a further excellent PowerPoint presentation relevant to his roles at NASA's Division of Space Biology, which, if time permitting, he may also be able to share with us. We've allotted an hour for your lecture, Barry, so it's your turn, and thank you again so much for substituting in at short notice.
It's no easy task to try to speak instead of Ray Kurzweil. So I hope you'll be tolerant of what I'm about to present. Also, I was informed about the need for someone to fill in a few hours before we were going to leave for Montreal. So I had to use an available slide set, PowerPoint set, and it's not exactly applicable, but I think has some parts of it have relevance to what we've been discussing. And if we can, I think, skip that first slide and go on to that. Well, we can skip that as well. Last year was the 40th anniversary of the first paper that we wrote on hepatitis B, and there was a kind of an event to commemorate that, a seminar and so forth. And the first time I sort of realized it was the 40th anniversary is when they asked me to come to the event. Now, what I'm talking about is a practical application of a scientific project, which initially started off with a basic science interest. And this is an example which I think is very common in applied science and applied medicine. When one is pursuing a particular fundamental question, and during the course of that, some application becomes obvious. So discovery of the hepatitis B virus did not start as a consequence of looking for the hepatitis B virus, but rather it was a consequence of an interest that we had, still have, in why some people get sick and others don't, a kind of a general issue. Many people are exposed to an infectious agent, and only some of them get sick. Now, why does that happen? Some of it's bad luck. Some of it's the environment that people are in. But some of it's due to inherited differences that make some people more susceptible to particular disease-causing agents, and makes others immune and renders others immune, or it has an impact on it. So we started out to look for inherited differences in serum proteins. Now, this work began in the 1950s, and it wasn't some years before the molecular biology happened, before we knew much about the genes or could manipulate and study them. So we were looking for variation at the phenotype level, that is, at the level of the protein. Much more difficult to do, by the way, than looking at variation and having polymorphisms at the genic level. It would take maybe 1 to 2 years to identify a protein polymorphism, and you can do a dozen in an afternoon using a genetic approach. By polymorphism, I mean genetic polymorphisms are systems that are much better known now because of the interest in localizing genes and the whole genome project. But these represent, they're loci in which there are multiple alleles, which may differ from each other only by a single amino acid, the product by a single amino acid or a small number of amino acids, but they actually have quite different functions. And it's the number of sites that are thought to be polymorphic is increasing, and I wouldn't be surprised that most genic sites are polymorphisms or some extension of that. That is, they may be kind of rare, very rare variants. Okay, so we started looking at serum proteins, and since we had reason to believe that these susceptibility genes would vary from place to place, depending on the environment and the disease hazards that were present, we spent a lot of time doing field work, going to places with very different environments and looking for variation in the proteins. So our first studies were done in Europe, actually, and then next study was done in West Africa, and then as a contrast, we went up to Alaska and looked at American Indians and Inuits, and then the Central Pacific and in Asia and so forth. So there was this wide variety of places that we studied. We found polymorphisms and very big differences in their distribution. Well, one of the techniques we developed was using the serum of people who had had many transfusions, with the expectation that if they received a transfusion of a serum protein, a variant that they themselves had not inherited, and if that difference was antigenic in character, they develop antibodies against it. So we looked for antibodies in people who had received many transfusions, 5, 10, 20, many more than that. And we tested them against proteins in normal individuals, or just anybody, in fact, usually from different populations. While using that, we actually did identify a protein polymorphism in the low-density lipoproteins that turns out to be related to Alzheimer's disease and to certain lipid dystrophies as well. But we also found an antibody in a transfused patient from New York City that reacted, not with very many people in the United States, but it did react with a serum in an Australian. And we subsequently found that the antibody that had developed in that transfused patient was directed against the surface of the hepatitis B virus. And the Australian, who had the antigen, which reacted with somebody 10,000 miles away, many thousands of miles away, was a carrier of the hepatitis B virus. Okay, so and then we spent a lot of time working on hepatitis B virus. but still maintaining this interest in the inherited variation. I mean, the question remained, why did the transfused person develop antibody and the person who had what we called Australian antigen, why did he become a carrier probably without disease? So it was interesting. The person who developed the antibody didn't have any symptoms of hepatitis, and the carrier, the person who was carrying hepatitis B virus, was probably asymptomatic at the time. So we identified a very seriously pathogenic virus by using the bloods of two people, neither of whom had hepatitis at the times the studies were done. Okay, now we switch, in a sense, to application, because we developed a vaccine quite soon after the discovery of the virus. And I'm going to talk primarily about applications. Now, first of all, HPV is a very common infection. This is kind of a review. I was told the way to give a talk is to tell everybody what you're going to say, then say it, and then tell them what you said. You know, so it's very common. It causes hepatitis, hepatocellular carcinoma, a very common cancer. The vaccine we developed is highly effective and wide use. There's been a dramatic drop in the number of carriers in the places where vaccination has taken place over the course of nearly 20 years now, more than 20 years. There's a significant decrease in hepatocellular carcinoma in certain places where the studies have been done. Then, quick switch. If this seems to be effective for hepatitis B virus and primary cancer, liver, very common cancer, there are many other cancers that are attributed to infectious agents, and then there are probably, and one is now in play, that is a papillomavirus vaccine and cancer of the cervix and certain other cancers. And there's evidence that this is effective in preventing those, and that's now the second cancer prevention vaccine. Then there's many other cancers. It's said about 15% of cancers are due to viruses. And there are probably many others in which the virus is part of the pathogenesis. Diseases rarely have one cause. There's always multiple causes. Usually you pick on one that seems somehow most fundamentally related to it. But in effect, what you want to find out about is the cause among many where you can do something about it. You can do something preventive. So there's evidence that this is preventive. There are multiple causes, and I recommended that there should be a priority on identifying these viral causes and doing something about it. I should tell you I'm having difficulty at my own institution, at the Fox Chase Cancer Center. It's not, has not been a popular field, but I think will become so. And again, we've gone to that. That's my team at its max when we had about 40 people in it. And they're the ones who worked on it. They all look kind of young and eager. They're all pretty old, and some dead right now. This was the original reaction that we saw between the antibody from the hemophilia patient from New York City and the Australian. This is a similar reaction. And that started the whole project. And again, it was a consequence of our investigation of inherited variation and how it relates to disease susceptibility. Okay, now this, we finally, after identifying this antigen-antibody reaction, we were able to look at the portion of the blood where the precipitin was. We looked at that arc, essentially. And this is a later image. But it shows a hepatitis B virus in the blood of carriers. First of all, there are vast amounts of these particles. And in some cases, they'll constitute 1 or 2% of the serum proteins. Could be made up of these related particles. Now, there are three kinds of particles. One is the whole virus, 42 nanometer, and it's got a surface antigen called hepatitis B surface antigen and a core antigen, and then the DNA is inside, and here's the core antigen. And then in addition to those, there are these small circular particles, these things, and the elongated particles. The small particles and the elongated particles are made up entirely of surface antigen. They don't have DNA, they don't replicate, they're not pathogenic. And it's these that are present in such high quantity. Now, I think perhaps just dwell on that for a bit longer. Now, we decided to develop a vaccine. And that was an interesting cause. Back in the 1970s, we were told that the feds weren't going to fund medical research as much as they had in the past, that they expected investigators to patent the stuff and or go into industrial use, manufacture. And it didn't happen, by the way. The funding continued to be very high for a while. But in any case, that spurred us to do something about it, showed you the motivation for doing that sort of thing. And we had found, using very insensitive methods, that we rarely saw somebody who both was a carrier of the hepatitis B virus, that has had all this material in their blood, so that if the same person would have the was a carrier and also had antibody against the HBsAg, against the surface. So we didn't see people in epidemiologic studies who were both had the virus and had antibody against it. That's consistent with the antibody against the surface being protective. One of the major problems for the finding of a vaccine for HIV and for hepatitis C virus, HCV, both of which are very common and very deadly diseases, but there's no vaccine for them. The problem is it's not known what's protective. Now, based on this very simple epidemiologic study, we had a very good clue that antibody against the surface antigen was protective. It was also the only antibody we knew at the time. So we applied for a patent in 1969 in which we isolated these surface antigen particles from the whole virus, from those particles, discarded the whole virus, treated the remaining surface antigen particle material with, in effect, very harsh methods that would kill any hepatitis virus, in fact, kill any known virus, and that was the vaccine. Now, that is and probably always will be a unique method for making a vaccine. It's a vaccine that's made from the blood of people who are carriers of the vaccine, of the virus. And the reason it probably won't be used anymore is because there aren't very many viruses that have this kind of combination of particles produced. And the other thing is there's a concern about using products derived from blood. But in any case, that vaccine was produced. We licensed a large company, Merck company. They did a very good job on producing this, upscaling it, and arranging for its testing. And it's been widely used for many years. And I think tens, maybe hundreds of millions of doses of this vaccine has been used. And at least as far as we can tell, based on numerous investigations, there have not been side effects, and it's highly protective. Now, just to go back there for a moment, these were some of the high points. The identification of the Australia antigen, recognized in '67 as part of the hepatitis B virus, the invention of the hepatitis B vaccine, which I've just mentioned, and then we looked into the question of whether hepatitis B virus caused primary cancer of the liver, and that was subsequently shown to be the fact. And then the high risk of carriers getting hepatitis, primary cancer of the liver, HCC, was demonstrated by a very excellent study, an epidemiologic study, again, done in Taiwan. And then there was kind of an agreement on the identification of the virus. And then in 1980s, there was a field trial done, by the way, with about, the final pool that was used for the study, the field test of the vaccine was about 1,000 people. Now, based on that study, the FDA approved the vaccine. The results were totally convincing, and Dr. Wolf Schmunis, late Dr. Wolf Schmunis, who organized the study, was very well done. And again, it was accepted. Just to remind you, the polio vaccine, the field trials for the polio vaccine involved 1.2 million children. There are trials set up now, even though the vaccine isn't available, for testing HIV vaccine, and that they've made provisions for studying tens of thousands of people. So Wolf Schmunis's trial was really quite remarkable to have been done with that. And so the WHO has made the point some years ago that this was the first cancer vaccine, which was very encouraging to look for others, and I think still is. Now, just to talk about the prevalence of these diseases, primary cancer of the liver, or hepatocellular carcinoma, it's the third most common cause of death from cancer in males worldwide, seventh most common cause of death in females worldwide. There's always a gender disproportion in anything that hepatitis B virus is involved with. There's about 1 million deaths per year, and about 80%, 75% is due to hepatitis B, and much of the rest to hepatitis C. And there are also many factors that are involved in pathogenesis. Now, hepatitis B vaccine was the first vaccine made by recombinant method. And it was, up until recently, the only recombinant vaccine, and one of the first of the successful recombinant products. And this was produced by several companies in the United States and elsewhere. Now, this is a diagram, a kind of simple diagram of the genome of hepatitis B virus. And for the moment, we'll just look at this gene S. And that is the gene that's got two start codons, and you get 3 pieces and various combinations of those, actually. That gene was removed and put in various cells, and eventually in yeast, and then going through fermenting process, the recombinant technique, and vast quantities of the vaccine are made that way. In one factory, you go in, three shifts, you can make all the vaccine you need in the world, probably. The price was terribly high to begin with, but there was competition in other countries. And the price is now very much lower. So one of the reasons why this was the first human virus that was sequenced, and that's because it has a very small genome, and this is a larger image of that gene. Now, looking at the epidemiology in the United States, there are about probably 2 million people infected in the United States with hepatitis B virus, and it varies a great deal from different individuals. Most of the people, there are about nearly 400 million carriers of hepatitis B virus in the world. About a third of the world's population has been infected with hepatitis B virus. That means they've been infected, and some of them, about 300 million, are currently infected, but others have been infected and developed antibody, and are, for the most part, their risk is very much less of getting the late consequences of chronic infection, that is, cancer of the liver and primary and chronic liver disease, debilitating chronic liver disease. But a third of the world's population has interacted with this virus. Now, the ability to become infected and to become a carrier, there's a genetic component. there are about 10 or 12 loci, polymorphic loci, that are known, and there are probably many more, in which at each of these loci, if you have a particular allele, then when you're infected with the hepatitis B virus, you're more likely to become a carrier of the virus. If you have alternative alleles, you're more likely to develop the protective antibody. Now, this isn't a single loci, it's multiple loci. And the effectiveness of the polymorphism probably varies from population to population. It's highly specific in that sense. Okay, so just as a reminder, most infectious diseases, most diseases are of this character, that they're related, that susceptibility is related to a genetic loci, to these polymorphic variations. So it's fair to say that for every disease, there's a environmental component, like infection with the hepatitis B virus, you have to get infected. There's a genetic component that has an impact on how you respond to the infectious agent. There's a behavioral component for many of these bloodborne diseases. If your behavior includes injection of drugs without properly sterilized needles, that behavior increases your probability, and a variety of others. You're all familiar with these from the what's known about HIV, whose method of transmission is very similar. The only hepatitis B is far more infectious than HIV, so it's more easily transmitted, actually. Okay, so again, I want to make that point very clearly, that when people say, well, this disease is genetic, that I think it's fair to say would be true for essentially all diseases. But it's not deterministic. If you take the vaccine, for example, then independent of your susceptibility status, you won't get infected. So, you know, behavior changes whether you're going to be, your fate's going to depend on your genetic makeup. And by the way, that's what medicine's all about. You know, we have certain genetic issues, and either try to decrease the probability of being exposed to an agent, or you treat it. So we're constantly changing the effects of genes. What's harder to change is behavior. Now, this again is the place where there's most hepatitis B virus and most primary cancer of the liver and most infection with the virus is China. And there are about over 100 million carriers in China. They have extremely high rates of primary cancer of the liver. And studies that were done on the island of Taiwan, there's about a 40% risk of carriers dying of cancer of the liver or primary cancer of chronic liver disease. And it's one of the major causes, or was one of the major causes of death in Taiwan in males. And actually in China, the impact of HPV is greater than that of HIV. Again, it's a big burden. It's a very hard disease to treat. There was recently a new chemotherapy introduced for cancer of the liver, which has now been accepted by the FDA. And the clinical trials show that the duration of life for the people who did not receive the treatment was like 8 or 9 months, and it was like 14 months for the treatment, which is better, but it's not a spectacular effect. On the other hand, prevention is much cheaper, much easier. Now, this is a compression of a very large number of studies, which have kind of simplified the data to show the prevalence of people who are carriers of the hepatitis B virus before the vaccination programs were instituted and afterwards. And in a regional study in China, it dropped from 16% to over 1%. Gambia from 10% to 0.6%. In Italy, from 10 to 0.8%, and so forth. So they're really striking things. Like in Alaska, the USA, Alaska, the acute cases dropped to 0. And I think, so there's been a profound change. Now, there's been a major change in the US population altogether in the acute cases, not the chronic cases, but that also is a result of the vaccination. 260,000 to 78,000 after. In Native Americans in Alaska dropped from 215, that's the total number, it's a relatively small population, to no cases. And very similar in Hawaii. And so there have been dramatic effects, and the vaccine's been in use about 20 years. And then there was also a study in Taiwan and another one in Korea in which the data was sufficient to arrive at this table. In Taiwan, the data of the cases per 100,000 in Taiwan was 0.7. By the way, this was in the impacted population. That would be in children who had been vaccinated and then looking 15, 20 years later. Now, there's a broad age distribution of cancer of the live in Taiwan, and the max would be about 50 years old. But you do get some in younger people. And it was possible to measure the change, and went from 0.7 to about half. And then in this other age group, 0.5 down to 0.13. And there was a similar study in Korea where, again, it went down from about 18.1. These were in adults per 100,000, down to less than 1 per 100,000. Again, dramatic change. This is quite a thorough study. This was a little less so. Okay, so that is a dramatic improvement in that, and the data for primary for cancer of the cervix is very similar. And we hope it's going to get even better as the vaccine is used more and more. So these are two really common cancers. Okay, now, having said that, there are about 400 million people in the world who are carriers of the hepatitis B virus now. Many of them in China and sub-Saharan Africa, but quite a few in the United States. By the way, primary cancer of the liver is one of the few cancers that's growing in incidence in the United States right now, probably because of Asian migration beginning some years ago, and they have a higher prevalence of no evidence, by the way, that they're spreading it elsewhere. But in any case, so there are all these carriers of the hepatitis B virus. We know they're at high risk. What can you do about it? Vaccine therapy, as far as I can tell, doesn't work. You know, treating people with vaccine. I know there's a lot of interest in doing it, and there's vaccines for treating cancers and so forth, and a great deal of research. But I think it's fair to say there aren't any widely used effective therapies for cancer using vaccines. And as a matter of fact, you might even say that for many other diseases. So although there have been attempts at it, there isn't data that treating with vaccine helps. Okay, so this was a study that was done by my colleagues at Fox Chase Cancer Center, in which they did a study in Hainan County, which is south of near Shanghai. They have very high prevalence of carriers there. And they followed these people for a long time along with their Chinese colleagues. And they looked at the mortality by the virus load at the beginning of the study. And this was a study carried on over 10 years, 11 years. So if you had very high titers of DNA in your blood, there was a very high probability of death. That is, survival was decreased. If you didn't have any measurable DNA, even though you were a carrier, which means the levels were quite low, the survival was much greater. And if you had sort of intermediate levels, then it was intermediate between those two. Okay, so that meant having high titers was bad news. Well, over the course of the years, and this is another indication that it indicates the baseline HBV DNA and the years of follow-up and the incidence. This is sort of the opposite of the other one. It has the incidence of liver cancer. So if you had really high titers, then you had a higher probability of getting cancer. If you had low titers, a low probability. Again, there were several studies that told you that addressed the issue of the relation of titer to outcome. High titer led to a bad outcome. And there have now been several studies using various drugs. Now, there are a dozen drugs for treatment of hepatitis B. They're sort of good but not great. And there have been studies done on probably the least effective ones, such as interferon, which is used very widely, and on lamivudine, which is a fairly new drug, about 5-10 years old, and on a drug that's been in use for about 3 years now, Baruch, entecavir. So these are pretty good drugs, but they're not fine. But there's now been data to show that if you treat these carriers early on and decrease the load, you don't even have to get it down to zero. It's very hard to decrease the level to zero because hepatitis B virus is integrated into the DNA of the host, and getting it out is extremely difficult. Okay, so even though the virus is still present, if you drop the levels down to about 5% of that would be in the levels of particles. It's much lower than in the placebo group, and this is a group that developed a mutation to a form of the virus, a viral mutation that increases the resistance to treatment. So that's intermediate, but still works. So the bottom line on this is that you can do a great deal to decrease the viral load and decrease the risk by using not such a great drug. You don't need a really good drug to do it. You need a drug that, obviously, the better it is and the more effective it is, and the less the possibility of developing tolerance to it, the better the drug is. Baruch has the advantage that it appears so far, anyhow, to have less development or resistance. And there's certain other drugs of that as well. So this means you don't have to have a perfect drug. And as a matter of fact, you probably want one that's not too effective because that has a greater or may have a greater probability of toxicity. Or you can use a pretty good drug and lower dose, so you can decrease the toxicity. Okay, so this is very encouraging. And what I'm trying to encourage, and many other people are, is for people to do more of this. And you can see that preventing, of course, is numero uno. A liver transplant, cancer of the liver is the main cause, is the main reason to do liver transplants. And they cost, well, a few years ago it was $500,000 a pop. I think it's probably more expensive now. And of course, the survival is much better than if you don't have the transplant, but it's it's still life-shortening. So it's much cheaper to prevent the cancer, either by primary prevention, which is the use of the vaccine, or secondary prevention, or secondary prevention/treatment by treating the people who are carriers but are asymptomatic. There are big issues. You have to decide which ones you're going to treat. People often get infected when they're born, and you want to be very careful about prescribing a drug to a newborn to use for the rest of his or her life. So there are issues that have to be resolved, but they're approachable primarily by appropriate clinical trials. So that's these are a list which I won't go through in any detail on the cancers that are thought, which are ascribed to viral causes. And there are quite a few of them. Some of them cause, pretty common diseases. And there's the papillomavirus, and there are undoubtedly others as well. So there are quite a few. And about 15% are caused by viruses, prevention by vaccination, or what we call treatment by delay. That is, these people in whom you load the titers, if they live long enough, they may get the cancer even with the, but nobody lives forever. So you want to treat them until, in effect, they die of something else, or live a normal lifespan. And there's an imperative to. Now, we're talking about a virus that's had an interaction with a very large number of the world's population. About 1/3, 2 billion people have been infected with it. The virus has very ancient antecedents. You can find it in animals other than humans, in mammals and fish, maybe in reptiles. Nobody's looked at things like bacteria and so forth. And so that means we've had a long association with this virus. We've lived it for very long. And it's slightly scary to think that the vaccination is now changing all that, is in the process of changing it. There's going to be consequences. Hard to know what they are. You can't tell the future. By the way, hearing the talks today reminded me of a statement I heard in a space seminar I went to, which had to do with kind of the future or prediction, what's going to happen. And the best statement I heard about that, if you want to predict the future, make it, which is what you're all about. Okay, now, the point that I'm making is that this has been around a long time. It's related to the genetics of the host, of the genetics of the person. That is, these genes that determine whether you're susceptible or not are part of your genetic makeup. And they're polymorphisms. That means in order to be maintained in the population, there have to be advantages and disadvantages. It's rare to look at advantages of infectious agents. But I should remind you that we have more bacterial cells in our body than we have our own cells. The GI tract, the respiratory tract, the genitourinary tract, they're all filled with bacteria. And we've been living happily with them for a long time. Now, many of those bacteria have phages in them, that is viruses, and they have a lot of phages compared to them. So there's even more viruses lurking around. They're I would say less than 1 or 2% of all the viruses, bacteria, and so forth are pathological. The reason we know so much about them is microbiology is medical microbiology. It started as medical microbiology, and therefore we kind of focus on the worst aspects of bacteria. But I can assure you the world would be a less happy place if we didn't have a lot of infectious agents. Having said that, I'm in the business of preventing infection and treating it when it happens. But I want to tell you about a very interesting non-pathological effect. And that has to do with, again, one of these observations that we made when we were doing the study in a place where there was quite a lot of hepatitis B virus. And we went to a small community in which there was a very high infection rate. And we studied the entire community. And since my initial interest was in genetics, that meant you looked at families. And we wanted to see how the virus related to survival and so forth. Well, one of the things we discovered was that the gender of offspring was related to whether the parents were either a carrier of the hepatitis B virus or had developed antibody against it. That's kind of strange. And first of all, we found that this was a very early study from 1960s, and this has been consistent. In practically every place we looked, the number of males who were carriers was higher than the number of females, with an exception here. And most of the studies since then have showed that you might say that hepatitis virus has a kind of bias, a favorable one in terms of females, that carriers are less common among females than males. But then in this study I just described, we looked at the whole community, pretty much, about 85% were tested. And then we looked at families where one parent or the other was a carrier. HBsAg positive means they carry the virus. And then in families where the parents were negative for the carrier, but one or the other had antibody against the surface. So these are the polar opposites in terms of response to infection. So when these people are infected, they became carriers, and these people are infected, they developed antibody. And these people didn't show any signs of infection. Well, the gender ratio, sex ratio, was significantly higher in the families where the parents were carriers than in those in which they had antibody, and those who seemed to be uninfected were intermediate. All right, we did this in 5 places, and the studies were consistent. And there were 1 or 2 studies that were done by colleagues elsewhere. But there hadn't been very much interest in this, and we sort of stopped working on it because there wasn't much more we could do, you know, at that point. We got on to other things like the vaccine. But we did a study in Greenland and Karkar, which is in Papua New Guinea. We did a study in Greece and the Philippines. We doing there. This was the second Greek study and one in France. And the data were all consistent. Well, not much happened, as I said, for a while. And then I had an email, and then I met a young woman who was doing her thesis in economics at Boston, at Harvard. And apparently she had read the book that I wrote about a scientific memoir. Her boyfriend had given it to her after some kind of argument. So it's a strange book to give as a gift after that. But nevertheless, she read it, and she came across a few pages where we discussed the gender difference. She got really intrigued by it. And she did several studies. And I'm showing you, they were very sophisticated. She's one of these, she's at Chicago now, very quantitative economist. In any case, she plotted, you know, if this gender ratio was prevalent, I think I have a few more minutes for the hour, isn't that right? Yeah, I'm just about finished. Then you'd expect the gender ratio to be more or less proportion to the number of carriers, percent of carriers in the population. So she plotted this in a variety of ways, leaving China out, putting China in. She was concerned about these outliers. But there's a linear relation between the rate of carriers, which she obtained from the literature, and the gender ratio at birth, which the World Health Organization keep their data on that. Okay, that's consistent with it. Now, there have been several places where the vaccination program has been spectacular. One of them is Alaska. Now, in Alaska, there's a high rate of carriers in Native Americans who are Indians, North American Indians. It's high there, but it's lower in Inuits and Eskimos, and it's lower in Alaskans of European origin. Okay, so the vaccination program conducted by the U.S. Public Health Service was incredibly good there, and they effectively vaccinated a very large percentage of the population. Okay, so if the vaccine decreased the number of carriers, you'd expect the gender ratio to go down in the places where there was a lot of carriers. Well, that was the case. Here's the Indian Native Americans, and over the course of years, after the vaccination began, the ratio went down, the males to females, but the other populations, it stayed more or less the same. Now, she did a more sophisticated study, actually, in Taiwan with the same general conclusion. As I said, her paper is a very sophisticated one. Okay, not only does that not imply, and it sort of is consistent, it's logical, that not only is your response to the virus related to the gender of your offspring, it causes it. It's the cause of it. So here we have a situation where a virus is involved in a non-pathological event, right? You can't exactly call the gender of your children a disease. And in a way, it's not surprising. When we've been interacting with this virus for a very long time, it's going to do a lot of things that we can't predict. And this is one of them. If, we have to, her paper caused a great sensation. It was covered in the Wall Street Journal, you know, and The Economist, you know, had a big thing on it. But in any case, I just want to conclude with one or two kind of general statements. One of them is animals, I'm going down to archaea, bacteria, viruses, are very variable. They differ a great deal. We're all a consequence of evolution. Now, one of the features of evolution is that you have to be prepared, a population or even a species or a genus has to be prepared for, in order to survive, has to be prepared for an unknown future and for an unknowable future, with the exception I just told you about making the future. You know, it's hard to predict it. And it's also, our present genome is a consequence of things that happened in the past. We share a third of our genome with archaea. Archaea are meant to be one of the very earliest forms, still very popular, still very common, very successful species. So that means all that stuff has come down to us, and we're using it. So first of all, the conclusion is nature isn't big into making new products. It likes to kind of recycle and use them for different purposes. But those old purposes are still there, and they're the preparation for the future. This also brings up the interesting idea that the past is a prelude to the future. I mean, the things that are going to adapt in the future, not all of them, but many of them will have been introduced a long time ago. We don't share all our genes, you know, with archaea. And as a consequence, living matter, humans, bugs, other animals, fish, birds, they're not perfect. And in fact, if anything's perfect, as I've said several times to others today, if something's perfect, it means by definition it's not perfect, because it means it's not ready for the future. Okay, engineers try to optimize function. That's not the way biology works. There's a big difference between, well, it may be a small difference, but the difference we don't know between a machine, like these wonderful robots we've been hearing about and something that's alive. And somebody hasn't figured that, we haven't really worked on that. Now, I was the director of the NASA Astrobiology Institute, and that was our goal, was to understand how life started and whether we share life with other places in the universe. Well, thank you very much for listening to that.